First question. Can you tell us about yourself and your background in the pharmaceutical industry? Answer. I may B.Pharm M.Sc chemistry graduate with a strong interest in pharmaceutical production and quality standards. Although I'm a fresher, I have studied key concepts like drug formulations, GMP, and safety protocols during my coursework. I'm eager to start my career in a well-established pharmaceutical company like yours and apply my academic knowledge in a real-time production environment. What do you know about our company and our products? Answer. I have researched your company's history, product portfolio, and recent developments. Your focus on sets you apart in the industry. I admire your commitment to quality and patient safety, as demonstrated by your adherence to GMP standards and successful product launches like. Additionally, your recent expansion into highlights your forward-thinking approach. What are the different departments in a pharmaceutical company? Answer. The major departments in a pharmaceutical company include production, for manufacturing medicines; quality control, for testing and verifying product quality; quality assurance, for ensuring compliance with standards and documentation; research and development, for developing new drugs; regulatory affairs, for handling government approvals; warehouse stores, for material storage; engineering and maintenance, for equipment and facility upkeep; safety health environment, for workplace safety and compliance. What is pH, and why is it important in pharma formulations? Answer. pH is a measure of the acidity or alkalinity of a solution, expressed on a scale from 0 to 14. It's calculated as the negative logarithm of the hydrogen ion concentration. In pharmaceutical formulations, pH is crucial because it affects the stability and solubility of drug molecules. It can influence drug absorption and bioavailability. It impacts patient comfort and tolerability, especially for injectable or topical medications. Many chemical reactions are pH dependent. Therefore, controlling pH is essential for ensuring product quality and efficacy. What are GMP and why are they important in pharmaceuticals? Answer. GMP stands for Good Manufacturing Practices. They are essential guidelines that ensure product quality, consistent production of drugs that meet predefined standards, patient safety, minimization of contamination risks and errors during manufacturing, regulatory compliance, adherence to FDA, EMA, or WHO standards for global acceptance, GMP covers all aspects of production, including raw material sourcing, equipment maintenance, employee hygiene, record keeping, and quality control processes. What do you do in case of gas leak or emergency situation? Answer. In a gas leak, I'd immediately alert personnel and evacuate the area according to established emergency protocols. I'd locate and activate the nearest emergency shut-off valve if safe. If trained, I'd use appropriate PPE and monitoring equipment to assess the situation. I would follow all instructions given by the safety officer and ensure that I am following the site emergency plan. I understand that safety is paramount, and I would always prioritize the safety of myself and others. What safety signs have you seen in pharma labs or plants? Answer. I have seen no smoking, flammable material, corrosive material, biohazard, and emergency exit signs. Also, PPE required, signs indicating mandatory eye protection, gloves, and lab coats. I am also familiar with signs that indicate the location of fire extinguishers, eye wash stations, and first aid kits. These signs are critical for preventing accidents and ensuring a safe working environment. I understand the importance of paying attention to all warning signs. What is a dosage form? Give examples. Answer. A dosage form is the physical form in which a drug is delivered to the patient. Examples include solid, tablets, capsules; liquid, syrups, solutions; semi-solid, creams, ointments; parenteral, injections. Inhalers and patches are also examples of advanced dosage forms. What is OS and how do you handle it during analysis? Answer. OS stands for out of specification, indicating a test result that doesn't meet predefined acceptance criteria. Upon encountering an OS, I would immediately stop the analysis, inform my supervisor, and document the observation. I would then follow the established OS investigation procedure, including checking equipment calibration, reanalyzing the sample, and investigating potential errors. Proper documentation and adherence to SOPs are crucial for a thorough investigation. How is a batch process executed and documented in pharma production? Answer. A batch process in pharmaceutical production involves batch definition, identifying the specific batch of product being manufactured. Process execution, following a validated procedure for each step, from raw material preparation to final packaging. Documentation, maintaining detailed records of each step, including raw material usage, equipment used, and quality control checks. Quality control, conducting tests at various stages to ensure product quality. Batch release, final approval for distribution after all quality checks are passed. Why is sulfuric acid used in pharma, and what precautions should be taken? Answer: sulfuric acid is used in pharmaceuticals for pH adjustment, cleaning processes, and synthesis of intermediates in drug manufacturing. For example, it's used in certain esterification reactions. Precautions include wearing full PPE, handling in a fume hood, slow addition to water, and proper storage away from incompatible materials. Due to its corrosive nature, any spills must be neutralized immediately with appropriate materials and disposed of according to strict chemical waste regulations. Can you explain the difference between QA and QC? Answer: quality assurance focuses on ensuring that processes and systems are in place to maintain quality standards. Involves setting policies, procedures, and training programs to prevent errors. Ensures compliance with regulatory requirements. Quality control concentrates on testing and inspection to verify that products meet quality standards. Involves conducting tests, sampling, and analyzing data to ensure product conformity. Identifies and corrects defects in the final product. QA prepares and approves SOPs, while QC tests the tablets for hardness and dissolution before release. What is a deviation? How is it handled in QA? Answer: a deviation is any departure from approved procedures or specifications. Example: a temperature excursion during storage. Deviations are documented, investigated, and evaluated for impact on product quality. Steps to handle it in QA. A. Immediately report the deviation. Do. Document it in a deviation form. 3. Investigate the root cause. 4. Implement CAPA. 5. Get QA approval to proceed or rework the batch. Example: if the temperature in the granulation area exceeds the limit, it's logged as a deviation, and QA investigates the reason before allowing further processing. What are CAPA and its role in QA? Answer: corrective and preventive action. CAPA is a systematic approach to identifying and correcting deviations or potential problems. Its role in QA is to ensure that corrective actions are taken to address identified issues and preventive measures are implemented to avoid future occurrences. CAPA helps maintain compliance with regulatory requirements by documenting and tracking all corrective and preventive actions. How do you conduct internal audits? Answer: internal audits involve a systematic examination of quality systems and processes against GMP standards and internal SOPs. Example: auditing the production area for compliance with cleanliness and documentation. I'd begin with an audit plan, review relevant documents, conduct interviews, and perform observations. Findings are documented as observations or non-conformances. A detailed audit report is generated, and follow-up actions are tracked to ensure corrective actions are implemented and effective. This process ensures ongoing compliance and quality improvements. What are impurities and how are they identified in drug samples? Answer: impurities in pharmaceuticals are substances other than the active ingredient or intended excipients. They can arise from starting materials, reagents, solvents, intermediates, degradation products, and excipients. Types of impurities: organic, inorganic, residual solvents, degradation products. Identification involves various analytical techniques. Chromatographic methods, HPLC, GCMS, and LCMS are commonly used for detecting organic impurities. Spectroscopic techniques, NMR, FTIR, and Raman spectroscopy help in structural elucidation. Thermal methods, TGA and AGA, are used for detecting residual solvents and other volatile impurities. Define boiling point and its relevance in process chemistry. Answer: the boiling point is the temperature at which a liquid turns into vapor under standard pressure. Relevance in pharma: helps select suitable solvents for synthesis and extraction. Used to control reactions. Important in distillation, drying, and crystallization. Example: a solvent with a low boiling point is preferred in drying because it evaporates quickly, speeding up the process. What are the different stages of a pharmaceutical product's life cycle? Answer: the pharmaceutical product life cycle includes drug discovery, identifying potential drug candidates. O. Preclinical development, in vitro and in vivo testing. O. Clinical trials, testing safety and efficacy in humans. O. Regulatory approval, submission and review by regulatory agencies. O. Manufacturing and commercialization, large-scale production and marketing. O. Post-marketing surveillance, monitoring safety and efficacy after release. O. Product lifecycle management, including patent expiration and generic entry. Explain the production changeover cleaning process and its importance. Answer. Changeover cleaning is performed when switching from one product to another in production to prevent cross-contamination. Steps include dismantling equipment, washing with appropriate cleaning agents, visual inspection, swab rinse testing, QA approval before restarting production. Example. When switching from tablet A to tablet B, all equipment is cleaned, tested, and cleared by QA to avoid trace contamination of ingredients. Can you explain the concept of bioavailability and bioequivalence? Answer. Bioavailability refers to the rate and extent to which the active drug enters the bloodstream and becomes available for action. Bioequivalence means that two drug products release the same amount of active ingredient in the body at the same rate and extent. Example. If a generic tablet shows the same blood concentration and effect as the branded tablet, it is considered bioequivalent and approved for substitution. What are the phases of clinical trials and their purpose? Answer. Clinical trials are divided into 4 phases, each with a specific purpose. Phase I, small group of healthy volunteers to test safety and dosage. Phase II, larger group to assess effectiveness and side effects. Phase III, large-scale testing to confirm effectiveness, monitor side effects, and compare with standard treatments. Phase IV, post-marketing studies to monitor long-term safety and effectiveness. Example. After Phase III success, a company may submit data to regulatory authorities for drug approval. What do you do if you find a mistake in a batch record? Answer. If a mistake is found in a batch record, immediate notification, inform the quality assurance team and relevant personnel. Investigation, conduct a thorough investigation to identify the cause of the error. Corrective action, implement corrective actions to prevent future occurrences. Documentation, update the batch record with the correction and maintain detailed documentation of the error and actions taken. Regulatory compliance, ensure that all actions align with GMP and regulatory standards. Example. If the recorded temperature during drying is incorrect, I would follow this process and ensure it's corrected per GMP guidelines. What's the purpose of validating equipment like FBDs and centrifuges? Answer. Validation of equipment like fluid bed dryers and centrifuges ensures that they operate consistently and reliably to produce high-quality products. This includes verifying operational parameters, demonstrating consistent product quality, ensuring compliance with GMP, providing documented evidence of equipment suitability. Validation minimizes risks and ensures product consistency. Example. Validating a fluid bed dryer confirms it dries granules uniformly at a set temperature and airflow. How is a centrifuge used in the pharma industry? Answer. A centrifuge is used to separate solid and liquid components by spinning at high speed. Uses in pharma. Separating solids from liquids. Clarifying solutions by removing particulate matter. Isolating and purifying proteins or other biomolecules. Separating emulsions or immiscible liquids. They are essential for purification and separation processes in API manufacturing and formulation. Example. In antibiotic production, a centrifuge separates the drug crystals from the fermentation broth. What are the major global regulatory bodies? Answer. Major global regulatory bodies include USFDA, regulates food and drugs in the US. EMA, regulates medicines in the European Union. KUTSCO, regulates drugs in India. PMDA, regulates drugs in Japan. MHRA, Medicines and Healthcare Products Regulatory Agency. WHO, World Health Organization. These bodies set standards for drug safety, efficacy, and quality, ensuring patient protection worldwide. Example. For a drug to be sold in the US, it must be approved by the USFDA. What are the types of reactors used in pharma industries? Answer. Common types include batch reactors, continuous stirred tank reactors, plug flow reactors, and glass-lined reactors. The choice depends on the process and product requirements. Example. For producing intermediates or IPI in small quantities, Glass-lined batch reactors are commonly used because they resist corrosion and are easy to clean. Explain the ANDA and NDA submission process. Answer. NDA, New Drug Application. Purpose: to obtain approval for a new drug. Process: involves submitting detailed information on the drug's safety, efficacy, and manufacturing process to regulatory bodies like the FDA. Content: includes clinical trial data, pharmacokinetic studies, and manufacturing information. ANDA, abbreviated New Drug Application. Purpose: to obtain approval for a generic version of an existing drug. Process: requires demonstrating bioequivalence to the reference-listed drug and compliance with GMP. Content: includes bioequivalence studies, formulation details, and manufacturing information. Example: after a branded drug patent expires, a company submits an ANDA to launch its generic version. Can you explain granulation and its types? Answer. Granulation is the process of forming granules from powder particles to improve flowability, compressibility, and reduce dust. Types of granulations. Wet granulation uses binder solution to form granules. Dry granulation compresses powders without liquid. Direct compression, no granulation needed. Powders directly compressed into tablets. Example: in wet granulation, starch paste is often used as a binder to form cohesive granules. How do you respond when a product is found out of trend? Answer. OT, out of trend means the result is within limits but doesn't follow the historical pattern. When a product is found OT, I would immediately document the observation and alert my supervisor. Initiate an investigation to identify the root cause, which includes reviewing batch records, testing methods, and equipment logs. Perform a risk assessment to determine the potential impact on product quality. Implement corrective actions and preventive actions to prevent recurrence. Example: if a tablet dissolution rate is OT, investigate equipment calibration, raw material quality, and process parameters. Name various manufactured dosage forms. Answer. Manufactured dosage forms include tablets, solid oral forms, often coated or uncoated. Capsules, containing powders or liquids within a gelatin or vegetarian shell. Injections, parenteral forms for direct administration into tissues or veins. Solutions, liquid preparations for oral or parenteral administration. Suspensions, liquid preparations containing undissolved particles. Creams and ointments, topical semi-solid forms for skin application. Patches, transdermal forms for controlled release through the skin. Inhalers, devices delivering drugs directly to the lungs. Suppositories, solid forms for rectal, vaginal, or urethral administration. What is serialization in pharmaceuticals and why is it necessary? Answer. Serialization in pharmaceuticals involves assigning a unique identifier to each product unit, allowing for real-time tracking throughout the supply chain. It is necessary for counterfeit prevention, helps prevent counterfeit drugs from entering the market. Product safety, enhances patient safety by enabling quick recalls if needed. Regulatory compliance, mandated by many countries to ensure traceability and authenticity. What are SOPs and why are they important in production? Answer. SOPs, standard operating procedures, are detailed written instructions that describe how to perform specific tasks. They are crucial in production because they ensure consistency and reproducibility. They minimize errors and deviations. They provide a training tool for personnel. They facilitate compliance with GMP. They create clear and concise instructions for workers to follow. Example: an SOP may describe the exact procedure for tablet compression, including machine settings, cleaning, and inspection steps. How do you manage reaction problems during synthesis? Answer. If an issue arises during chemical synthesis, stop the reaction if it's unsafe. Check parameters pH, temp, pressure, time. Inform senior chemist or production head. Review the batch manufacturing record BMR. Conduct root cause analysis and document the deviation. Apply CAPA before resuming production. Example: if reaction turns darker than expected, it may indicate overreaction or contamination. It must be reported immediately. What is the purpose of SS300 solar grade steel testing in pharma equipment? Answer. SS300 stainless steel 300 is commonly used in pharma equipment due to its corrosion resistance and durability. Purpose of testing: ensure material compatibility with chemicals and solvents. Prevent metal contamination in products. Comply with GMP and material standards. Verify passivation and surface finish quality. Example. SS-3 is used for reactors, pipelines, and vessels handling acidic or high-purity materials in cleanroom production. What are contamination and cross-contamination? How can they be prevented? Answer. Contamination is the unintended presence of impurities in a product. Cross-contamination is the transfer of material from one product to another. Prevention methods. Follow proper cleaning and sanitization. Use dedicated equipment or clean in between products. Implement line clearance. Maintain personal hygiene and area segregation. Example. Using separate rooms for penicillin products prevents cross-contamination of non-penicillin drugs. How is warehouse material stored and managed in pharma? Answer. Pharma warehouse follow GMP storage practices, including material segregation, raw materials, IPIG, excipients, and packaging materials. Labeling, status labels. Environmental control, temperature and humidity monitoring. AFIFO or FIFO principles. Access control to avoid unauthorized entry. Example. Temperature-sensitive products like vaccines are stored between 2 degrees Celsius, 8 degrees Celsius in cold rooms. What is the difference between a generic drug and a branded drug? Answer. A branded drug is the original patented product developed and marketed by a pharmaceutical company. A generic drug is a copy of the branded drug with same active ingredient, strength, dose, and bioequivalence. Example. Branded, Augmentin. Generic, amoxicillin plus clavulanic acid by a generic manufacturer. What is line clearance? Why is it done before starting a new batch? Answer. Line clearance is the process of thoroughly inspecting and cleaning production lines and equipment before starting a new batch. It's done to prevent cross-contamination. Ensure all materials from the previous batch are removed. Verify the equipment is clean and ready. Document the clearance for GMP compliance. It is a vital step in ensuring product quality and patient safety. What are the steps for material handling and dispensing as per GMP? Answer. 1. Receive materials with proper documentation. 2. Check material status. 3. Store in designated area under controlled conditions. 4. Weigh dispense materials in a clean area as per BMR. 5. Label and record batch numbers and quantities. 6. Transfer to production securely. Example. IPIs are dispensed in weighing booths with proper gowning and dust control, and each container is labeled with usage quantity. How do you clean reactors between batches CIP-SIP? Answer. CIP clean in places automated systems with cleaning agents and water to clean internal surfaces. SIP sterilize in places steam or chemical sterilant to sterilize the system without disassembly. Example. After completing a paracetamol batch, the reactor undergoes CIP with hot water and detergent, followed by SIP using steam to ensure no microbial contamination before the next batch. You observe temperature rising abnormally in a reactor, what steps would you take? Answer. Immediately stop heating, check cooling system function, reduce agitation if needed, alert the control room, and follow emergency shutdown protocols if the temperature continues to rise. Example. During an exothermic reaction, if the temperature goes above the set limit, I would shut down the heating coil, activate the cooling jacket, and inform the supervisor immediately for further action. What are common issues faced during reactor operation, and how do you troubleshoot them? Answer. Issues include temperature control failure, poor mixing, pressure buildup, leakage, and mechanical seal failure. Troubleshooting involves monitoring parameters, checking equipment integrity, validating control systems, and following SOPs for maintenance and repair. Example. If mixing is poor, it might be due to a damaged agitator blade. I would pause the batch, inspect the agitator, and coordinate with maintenance for replacement.